Assalamualaikum and hi students. So today we are going to discuss the questions from P4S 2. Question 1. Which of the following genotypes is homozygous dominant? The answer is B. X capital A, X capital A. This is because the option A is homozygous recessive, while C is hemizygous recessive because it's only have one X. And the last one is heterozygous. Question 2. An alternative form of a gene that may produce distinct phenotypic effects is known as, the answer is allele. Locus is the position of the allele on a chromosome, while genotype is the combination of alleles such as capital A, homozygous dominant, capital A, small a, heterozygous, which will give the dominant phenotype. For this example is brown eye color. The other genotype is homozygous recessive, which is small a, which will give out the recessive phenotype blue eye color. Question 3. Identify the type of cross that occur between plants of same generation with same characteristic. The key point is same generation. The answer is self-cross. For example, heterozygous cross with another heterozygous will give out the phenotypic ratio 3:1, while for test cross, for example, heterozygous cross with homozygous recessive will give out the phenotypic ratio 1:1. Question 4. Which of the following characteristics of pea plants were used by Mendel in his experiments? The answer is A, 1 and 2 only, because among the 7 characteristics observed by Mendel, pod length is not one of them. Question 5. If a true breeding tall pea plant is crossed with a true breeding short pea plant, the F1 generation will be all tall plant. When the F1 is self-fertilized, the phenotypic ratio of F2 will be, the answer is C, 3 tall and 1 short pea plant. This is based on the genetic diagram shown on the right. Question 6. State the genotype that would result from the cross between homozygous dominant and heterozygous. The answer is B, 1 and 3 only. So based on the genetic diagram, it is not possible for you to get an offspring with homozygous recessive. Question 7. Which of the following ratio will be the result of a dihybrid test cross? The answer is D, 1:1 to 1:1. For A, that is the ratio for monohybrid test cross, while B is the ratio for monohybrid self-cross. C is the phenotypic ratio for codominance and incomplete dominance self-cross. Question 8. Q square in Hardy-Weinberg equation indicates frequency for individuals D that are homozygous recessive. This is based on the equation P square plus 2PQ plus Q square equal to 1. P square is referring to the frequency of homozygous dominant genotype, while 2PQ is referring to the frequency of heterozygous genotype. Both P square plus 2PQ will show dominant phenotype. The only recessive phenotype is Q square, which is referring to the frequency of homozygous recessive genotype. Question 9. Which of the following situations unfollows Hardy-Weinberg equilibrium? The answer is B, mutation. This is because when mutation occurs, allele and genotype frequency may change through the loss or addition of alleles. Question 10. Which of the following assumptions will not change the allele frequencies in a population? The answer is A, 1 and 2 only. Why? Because genetic drift will slowly change the allele frequencies in the population. Question 11. Transfer of information from DNA to RNA is known as transcription. The answer is B. This is shown as in the figure on the right side. The process comprises of initiation, elongation, and termination. Question 12. What is the function of replication? The answer is A, to synthesize DNA. This process occurs during S phase of interphase, where the chromatids will become sister chromatids. Synthesizing mRNA is a process of transcription, while synthesizing protein is a process of translation. Question 13. Where does DNA replication occur? The answer is nucleus, as shown in the figure. Nucleolus is the dense, dark area of chromatin involved in making rRNA for the synthesis of ribosome, while cytoplasm is the jelly-like structure outside nucleus. Nucleoplasm is the fluid in the interior part of nucleus containing chromatin. Question 14. Choose the components of operon. The answer is A. As shown in the figure, like I is not part of the operon, but it controls the operon. It is the regulatory gene. Question 15. Which of the following is false about the enzymes involved in DNA replication? The answer is B. DNA polymerase 1 does not add DNA nucleotides, but it removes and replaces RNA primers with DNA nucleotides. Question 16. Which flow show the concept of central dogma in biological system? The answer is A. DNA will undergo transcription to produce mRNA, and that mRNA will leave nucleus, go to the ribosome to undergo translation process to produce protein. Last question, question 17. Which of the following enzymes are involved in DNA replication? The answer is B, 1 and 3 only. Why? Because RNA polymerase is involved in transcription, not DNA replication. In DNA replication, there are 6 enzymes involved, which are helicase, topoisomerase, primase, DNA polymerase 3, DNA polymerase 1, and DNA ligase. Other than that, we also have proteins, single-strand binding proteins. Alhamdulillah, we finished discuss all the questions in P4S 2. So, thank you for your attention and I wish you all the best for UPS 2. Bye. Assalamualaikum.